Good afternoon, everyone. So, in continuation, we will discuss more about the basic of an electron microscope, cryo-sample preparation, and data collection. So, next slide.
The next one. Okay.
Yes. Next
Okay.
slide, sir, please.
Next, just 1 minute. Actually,
Yeah.
just trying to.
Okay.
Okay.
So, generally the most common instrument in any life science laboratory is a microscope. And whenever we heard the term microscope, the another term used to come in our mind is the resolution. So the resolution is the distance at which we can see two objects is at two different entities. So if we talk about the resolution of our eyes, it's usually 0.2 mm. And if we wanted to see anything which is having size less than 0.2 mm, we need any kind of help that may be a microscope. And if we talk about the resolution that we can achieve by using the visible light is somewhere limited with 200 nanometer, which is approximate half of the wavelength of visible light. And if we wanted to study the at the if we wanted to study or visualize the particle at atomic resolution, we need something which is having wavelength in the same order. Next.
Okay. Okay. I think I'm not able to. Okay. Yeah, you can start.
Okay. So briefly we will discuss about the history of microscope, how it developed, and how this field of microscopy evolved over time. So we can trace the history of microscopy back in the early 40th century when the spectacles first made in the Italy. And in 1590, a father-son duo made the first microscope. And we all know that the Robert who published his Micrographia, in which he observed particles, several particles or several small objects, several particle or several small objects, and Anton van Leeuwenhoek came in limelight when he first observed the bacteria inside the microscope. And similarly, after that, Ernest Abbe gave a limit of resolution and derived a relation between the wavelength and linking the resolution with the wavelength of source of illumination. Then in 1897, J.J. Thomson discovered the electron as a subatomic particle. Now in 1924, de Broglie derived a mathematical equation which stated that we can change the wavelength of electron by changing the R accelerating voltage. So it means if we use higher accelerating voltage, we can decrease our wavelength or and increase our resolution. And in 1926, Bosch made the first electromagnetic lens. Made the discover that we can focus or deflect the electron beam by using electromagnetic lenses. And in 1929, Ernest Tuska completed his PhD on electromagnetic lenses.
Next slide.
Next slide. And now the field of electron microscopy began when first prototype of TEM was developed in 1931, and in the concept of single particle analysis was started in somewhere in 1975 when the 7 angstrom electron density map was prepared by using single particle analysis for protein bacteria rhodopsin. And in 1981, the process of vitrification was developed, After that, the same protein again studied, and we got resolution of 3.5 angstrom. And in 2008, somewhere, the direct electron detector revolutionized the field of microscopy. And we all know that in 2017, the Nobel laureate got no, they got the Nobel Prize by uh, in the cryo-electron microscopy by providing the subatomic resolution structure. This is all. Next person. So this is the basic anatomy of both optical microscope and electron microscope. Both are made up of in a similar way but having different source of light. As in light microscope, we are using light as a source of illumination, while in electron microscope, we are using electron beam as a source of illumination. In light microscope, we are using glass lenses to focus our beam, while in electron microscope, we are using electromagnetic lenses. In optical microscope, we can observe image directly with our naked eyes, while it's not possible in EM to observe the image directly with our eye because they are high energy carrying particles. Now we will discuss about how the electron interfere.
Hello?
Yes, sir. Is there something?
No issues, no issues. Okay.
Oh.
Yes.
So when our incidence beam fall on our object, it may got transmitted or scattered back. So it may transmitted by losing some energy, which we call inelastically scattered electron, or may transmitted without any interaction, or may transmitted with a little bit interaction but without losing energy, or they can backscattered, which generally this backscattered electron we are using for image creation in scanning electron microscopy. Correct? They the backscattered electron release some kind of X-rays, which are generally we use for elemental detection and Auger electrons and other things. So if we talk about the transmission electron microscopy, elastically scattered electron and the direct beam are of major interest. Next slide, sir. So this is the basic anatomy of a normal TEM. Over the top, we are having illumination system, which include mainly the source of our electron. After the condenser lenses and condenser lens aperture. Now come to the image formation part, which is mainly include objective lenses, our goniometer, and then objective lens aperture. And the magnification part is carried out by the intermediate lenses and projector lenses. And in last, we are having our detector. It may be direct or CCD. Next slide. So if we discuss a little bit about the source of electron, we can generate a coherent beam of electron either by using thermoionic emission or by using electromagnetic field. So basically we as a source of electron we are using tungsten filament or lanthanum boride filament, hexaboride filament, or maybe PEG emission, field emission gun. So to emit the beam from this tungsten filament, we have to maintain it at very high negative potential. For example, we can say that if our microscope is 200 kV, it means the difference between the potential between our cathode or our gun to the our anode is 200 kV. So this is our accelerating voltage. Higher the voltage, lesser wavelength we can get and higher resolution we can achieve. So in tungsten filament, we pass current through this filament, and due to this current, this filament got heat, and the electron which get energy by this heating emerge out greater than the work function of tungsten emerge out from the tip of our filament and captured by this V0 cylinder, which is maintained at a further lower voltage, which is called the difference between the voltage of a tungsten filament and this V0 cup is called bias, and then further accelerate with the help of our accelerating stack or anode. If we talk about the lanthanum boride hexaboride gun, or it is also based on the thermoionic emission. It means we heat our filament and it will lead to the emersion of a coherent beam. It is made up of single lanthanum boride crystal, and it is much more coherent than the tungsten filament, having less energy spread as compared to tungsten filament, but it is little bit more costly. Mostly in cryo-EM, we generally use field emission gun. Either it may be Schottky or it may be cold field emission gun. So it is made up of single tungsten crystal, and we can lower down the we lower down the work function of tungsten by coating it with the zirconium oxide, and it is having very fine tip or hair and lead to generation of very much coherent beam and with very much less energy spread as compared to the lanthanum hexaboride or tungsten filament. But the main reason that everyone is or every microscope is not having is that it is much more costly and need much more maintenance. Next slide. Next slide, please. Next one is the comparative chart of the different source of electron, what is their benefit, and their pros and cons we both are indicating here. So in normal tungsten filament, we have to heat up to 2700 Kelvin. In lanthanum boride, beam produce beam of electron produced at around 100 Kelvin, and in Schottky up at 1800, and while in cold field emission gun nearly at 300 Kelvin of heating. But the main difference is in brightness and the chromatic aberration or the energy spread. Tungsten filament give poor brightness and having high chromatic aberration, which means our resolution is compromised here due to chromatic aberration. But the benefit is that we can operate it at a optimal vacuum. It doesn't require very high ultra vacuum condition inside the column. Next. If we talk about the functioning of an electromagnetic lens, how it used to shape our electron beam on focus, it acts in a similar way like our normal glass lens. So suppose if our source of electron is here or our object is here, and some of the electron which pass through the center of the lens doesn't reflect or disperse or tilted. So they focused on the focus point. But our main concern are those electrons which are far away axis, then how to focus them. So the electromagnetic lenses are made up of bundle of copper wire which is having current, which current is continuously flowing through them and which result into creation of a magnetic field. When a moving electron enter into this electromagnetic field, it field force which result into the focusing of this electron onto the focal plane. Next. So a basic lens system, either it is condenser lens or objective lens or projector lens, having mainly four components. First is the deflector, then lens, then stigmator, and then aperture. Electromagnetic lenses having a stack of different electromagnetic lens over it, and it is not possible to align all the electromagnetic lenses very precisely. So when we are observing or our image at much higher magnification, a little bit of difference can give us a big effect. So to instead of aligning all the electromagnetic lenses much more perfectly, we can introduce a deflector pair. These are nothing but the electromagnetic coil which deflect our beam and make it towards the center or of our electromagnetic lenses. Then come the stigmator. There may be the difference in the strength of electromagnetic field around the coil of copper wire or around our lenses, which result into the focusing or dispersing of some of the electron before the focal plane. So for that, a pair of stigmators are placed below the electromagnetic lenses. And to limit the electron which are far away from optical axis, we used aperture generally to cut out those electrons. Next. So the first lens in our electron microscope is condenser lens. It is placed just below the accelerating stack or argon, which is having filament, V0 cylinder, accelerating stack, then come to condenser lens. So condenser lens receive beam from the electron gun and guide it to the sample or object. Generally, a combination of two lenses along with a condenser lens aperture we used. First, lens aperture, or we also used to call it sport size, create a demagnified image of the gun crossover and to control the sport size. The second condenser lens affects the convergence of the beam at the specimen, and the diameter of illumination area can be controlled by it on the specimen. So another and the most important lens in an electron microscope or in any microscope is the objective lens. It is the lens where our first image is formed, and the objective lens collect the electron which are transmitted from the our object and then focused them to the back focal plane. To limit the electron which are far away from our optical axis, we used here the optical aperture. Next. Now come to the intermediate and projector lenses. So intermediate lenses uh, generally depend upon the what magnification we are visualizing or doing our imaging. They switch on and off accordingly, and the multiple intermediate lenses we are using in a microscope, and the projector lens is the final lens in the column that project the image onto the viewing screen or camera. Next. Now come to the lens aberration or lens imperfection. First, we already talk about the stigmator, which is used to reduce the astigmation, mainly the aberration or the imperfection which caused by the uneven field of the of an electromagnetic around an electromagnetic lens. Another is chromatic aberration. Although the electron source that we are using are coherent in both manner, either it's spatial or both temporal. But after interaction and or due to some poor vacuum or another thing, we got chromatic aberration also because the electron which lose energy you used to disperse more and focus earlier than our focal plane. Then come to spherical aberration. This is a similar kind of aberration that we are dealing in the optical microscopy with the glass lenses that are the edges of our magnetic lens used to bend more. So the electron which are passing through the edge of our electromagnetic lens that used to focus before our focal plane, which leads to the blurring of our image. So we can control or manage this by using the aperture or limit that portion which are coming from the edges of our magnetic lenses. So if we take the overview of the normal time, top of the instrument we are having filament which used to generate the coherent beam. Below that we are having p-naught chamber which used to shape our beam that is emerging from the filament. Now the accelerating stack which used to accelerate our electron. For example, if we are saying our microscope is of 200 kV, so we are saying that this the difference between the voltage between the filament and the lower accelerating stack is 200 kV. So next, no, sir, previous one. So now come to the condenser lens system, which is having gun deflector here because above that we are having gun. So the beam which might not be at the center of the lenses, so we can make it center by using the gun deflector. And the key that we are using in on our microscope are called gun shift or gun tilt. So this is the first lens in the condenser lens system, CL1 or called sport size, and another one is the CL2. By increasing or decreasing the current in it, we can control the intensity of our beam, and these are called condenser stigmatism, then called condenser aperture. If we talk about the objective lens system, here the beam con- pass by the condenser lens system and pass through the object, and image is created here in the objective lens system and pass on to the our different intermediate lens and projector lenses, and then finally are detected down. So next. So if we talk about the detector, mainly we can categorize detector into two types, either CCD or CMOS based. In CCD detector, we are having a scintillator layer which converts the bombarding electron into photon, and then these photons are captured by the optical fiber below it and then converted into charge below it. But this the detective quantum efficiency of the CCD detectors is very low, and it may result into less signal to noise ratio. And in case of direct detector, we are having CMOS detector here, which directly convert the electron which are coming from our projector lens to charge. and help us in achieving better resolution. Next step. Now come to the energy filter. Energy filter means, as we studied earlier in a slide, the electron matter interactions. So the electron which lose energy during the interaction with the object, we want to filter them out from those which are elastically scattered. For that, we are using energy filters. They can be in-column energy filter or post-column energy filters. In-column energy filters are also called the omega type. In-column energy filters are placed uh, before the projector lens or between the objective lens and intermediate lens, while the post-column energy filters are placed below the our projector lenses. They are nothing but the electromagnetic lenses or the prism, which used to allow only the electron which are scattered elastically or which lose energy either get absorbed by the dispersion by caused by these lenses in the wall of the microscope or limited by slit. Next. So if we talk about the sample preparation workflow, so instead of directly going to cryo-electron microscopy for single particle analysis, it's better to go to the first to the room temperature temp by using negative staining, and then we can go to the cryo visualization, grid optimization, and we can collect the data then, and we can construct single particle structure. Next. So to do negatively staining, generally we use uranyl acetate, phosphotungstic acid, or ammonium molybdate or uranyl stain as to stain our sample either negatively or positively. Next. So to place our sample or to put our sample inside the microscope, we need some kind of support. Like in light microscopy, we are using glass slide, but in cryo-electron microscopy, it is not possible to use glass slide. So we used a metallic sport, which we called grid. It can be made up of either copper, nickel, molybdenum, or any other metal, or maybe gold. So if we talk about the grids which are having square pattern, it either it can be have 200 squares in it, 300 or 400, and much more. If they are having a single slot or a single notch inside the grid, we generally use this kind of grid in ultra section. And suppose if we wanted to locate our sample at a particular area and then again go back to imaging in some different microscope or having higher configuration, so we can use finder grids. So over the this framework of a metal, we are having a coat of a thin carbon film or maybe formyvar or maybe both. So usually this film is generally 3 to 10 nanometer based on our uses, or the size of this grid is usually 3.05 mm. So the carbon coated grid may be of different kind. It may have a continuous carbon over it, and it may have hole in it. Used to call it holy grid. And if the holes are arranged in a particular pattern, we called them quanticoil grid. And these type of grid, quanticoil grid, help us in automation of our data collection in Cryo-EM. Or they may be lacy grid, where the holes are cut in an irregular manner and having irregular size and shape. Suppose if we wanted to purchase the grid for our uses. So this kind of things are written on it. The copper 300 carbon. It means the PAX code or the framework is made up of copper having 300 meshes in it or square in it and having a layer of continuous carbon over it. And like copper 300 quantifoil R 1.2/1.3 means the basic framework is made up of copper metal having 300 squares and having quantifoil layer over it. The each hole having a radius of 1.2 micron meter and the distance between two hole is 1.3 micron meter. Next slide. So when we come to the protocol of negative staining, so as we discussed earlier that our grid used to have a thin layer of carbon which is generally hydrophobic. So to spread our sample properly on our grid, we have to do flow discharge or we have to make it hydrophilic or we can um, clean out the other impurities which are on our grid during the preparation by using the flow discharge. So first we flow discharge our grid, then within maximum I can say 1 hour we have to put our sample on it, and generally we used to incubate for 1 minute for it and then blot the extra sample with the help of a blotting sheet and then put our stain. Generally we use 1 to 2% of PTA in our lab for it and then incubate for 30 to 40 seconds depend on our mm, particle type or we have to still dry accordingly, and then after drying we can go for imaging in room temperature time or next. So what if we wanted to see the intracellular structure? It is not possible to see in the intact cell. For that we have to prepare the ultra thin sections. For doing this, first we have to fix our sample, uh, generally by using a mixture of glutaldehyde and paraformaldehyde, which are prepared in zero point 0.1 molar of phosphate buffer for 12 hours at 4 degrees Celsius. The duration we can change according to our ty- type of sample. For example, cells need a less duration while the tissue need more duration for fixation. And then we can wash out this primary fixative 2 to 3 times by using zero point 0.1 molar phosphate buffer. Then come to the secondary fixation, which generally we use to fix the lipid in our sample, for which we are using 1% osmium tetroxide, which generally provide the black color to our tissue or our sample or cell entry. Then come to washing to wash out the osmium tetroxide. We generally use to wash with 2 to 3 times with 1.1 molar phosphate water. So to provide the support for ultra sectioning, we have to embedded our sample into a resin, either it may be epoxy or araldite. So for proper embedding, we have to dehydrate our sample in a step-by-step manner, which we can do by 30% first, then 50 followed by 50%, 70, 90, and then finally by absolute. We can do either by using the ethanol or by using the ethyl. Next, next slide. Please. After the dehydration, we have to embedded our tissue or cells inside the resin to provide a firm support during cutting. So here we gradually add on the resin along with the ethanol. Like initially in 1 is to 3 ratio, then 2 is to 3 ratio, and then 3 is to 1 ratio, and then pure resin. So before to polymerize that resin, we have to put our sample in last at 70 degrees Celsius for generally for 16 to 24 or 48 hours. So we put our sample inside a mold or a capsule and then pour the 100% resin and then put into the oven at 70 degrees Celsius for 2 days. Then come to the ultra sectioning. So we are having our block ready for cutting. So first we have to chop the surface of the block to made a trapezium, which is generally usual 1 mm in size. And then by aligning our ultra microtome along with knife edge, we can do cutting. And by getting a golden section or grayish transparent section, it means the R sections are generally below 80 nanometer in thickness. We pick them out with the help of the loop and place in on our grid, and then after staining we can check our ultra sections in the microscope. Generally, in case of ultra sectioning, we used to do at lower accelerating voltage. It means either if suppose we are having 120 kV accelerating voltage, then it is there is a more probability that our sample melt down or our resin melt down before imaging. So we generally used to do at 80 kV at lower accelerating voltage. So here is we are representing our mold that we are using for embedding our sample, and we can also use the capsule one, that is second one, and these are the block which are like kind of that thing is represented our sample. And this is upper one is having a glass knife attached along with a board to collect our sample. So this is the alignment. Here is we are having our cutting arm or our lever which is holding our sample. Below that we are having a glass knife attached with the board. By moving this knife along with the edge of glass knife, and we can get our pin section. So this is the picture of our instrument that we are having in our laboratory, like EMUC7. It is also having the cryo attachment. So here we can also do the cryo section. Next. So now come to our main part. Why do we use Cryo-EM when we are already using negative stain? So in negative stain, there are lot of artifacts either due to stain or due to dehydration. We can't preserve our sample in native state in negative staining. So Cryo-EM provide edge over all these requirements. Then we can achieve a high resolution by using Cryo-EM by preserving our sample in native state and can construct single particle structure. The main requirement for cryo-EM is our, we can use sample concentration somewhere between 10 nanomolar to 10 micromolar, depending totally on our molecular weight or our sample type. And generally, we put 3 microliter of sample per grade. And if sample is not too homogeneous, then it can be okay with our cryo. So this is the basic workflow from MCT to the 3D structure for single particle analysis. We put our sample on the grid, then plunge it inside the liquid ethane, observe inside the microscope, collect large number of data, process them, align them, and construct our 3D construction. Next. So this is the CP3 plunger assembly that we are having. Top of it, we are having display unit, which indicate the temperature of ethane cup. We can set our plunging time, and the lower black window represent the humidity that we are having inside our humidity chamber. This upper one is the sample holding or tweezer holding rod, and the both of the side we are having plunging arm having bloating sheets over, and below that we are having workstation. So The first step in the preparation of our sample is we wet our humidity rod and put it inside the humidity chamber. And after the humidity reach around 80 or 80 above, we start our sample preparation. Meanwhile, we can pre-cool our workstation and fill the liquid ethane inside it. Next, so once our condition reached, for example, we set here the plunging temperature at minus 171 time 3 seconds, and our humidity reached at 80 above 80%, and this 22.3 is the ambient room temperature that we are plunging at. So this is the processor. We change the bloating sheet of our plunging arm that we are having open nitrogen cylinder to provide the force for plunging. Next. And this is black rod is the grid transfer tool that we are using to transfer our grid holder box from plunging assembly to loading assembly, and we put the our grid tool holding box with the help of grid transfer tool inside the our workstation, which is having ethane cup and a blotting sheet to blot out the extra ethane. After reaching all the conditions like humidity and temperature, we blow discharge our grid and hold with a special kind of tweezer which is meant for this purpose only, and then put the tweezer inside the chamber with the help of that rod that I earlier showed. And then next, we load our sample or that grid and then rotate the tweezer parallel to the blotting arm and then press a start button which provide force to the plunging arm and then blot our sample here. Next, and very suddenly it goes deep into the ethane cup. So here we blot the extra ethane over that filter paper we earlier put, and then transfer our grid to the grid holding box, and then with the help of grid transfer rod either we can store it and we can load into our cryo holder for imaging. So this is the assembly that we are using for loading our sample inside the holder or the cartridge of the holder. We are having 626 and 911 holder here, and so before loading our sample inside the holder, we have to pre-cool this assembly, which is having diwar. We generally used to maintain the temperature of the tip of the holder where we are going to load our sample. Here we transferred our sample from the grid box to that notch that is shown in the picture with the help of tweezer and secure it with the help of a strip ring. Next. Then we are having side entry microscope not with the auto loader, so we have to load manually. So if we insert our sample or with the help of this cryo holder inside the microscope, and after stabilization generally 10 to 20 minutes we can visualize our grid. So during visualization we select a proper area which is having optimal thickness of eyes. Then after doing initial alignment we can start imaging like aperture insertion, z height adjustment, aperture centering, and objective axis centering, and setting our defocus values. Next. So During screening procedure, we generally use to collect data manually by using manual MDS. MDS means minimal dose system. We generally use to mm, move between the search, focus, and photo mode. And with the help of quantifiable grid and different software, we can automate our collection also. So here in our facility, we are having GEOL 2200 FS microscope. So along with this, they provided an software for automated data collection, which is called TEDAS, GEOL Automated Data Acquisition System. So like So the first step in this automated data selection software is this: we select our area with the optimal eye thickness and the particle distribution. Then we select our recipe. Recipe means generally protocol, which is having how at what magnification we want to collect our data, how many number of frame we are going to collect, at what defocus value we are going to collect, and other things. Then we have to do basic alignments, and this then they prepared a global map or the montage by taking small picture, by taking picture at very low magnification, usually at 50 meg. And then we select our desired square for the data collection and calibration. Then we have to align the photo mode, focus mode, and search mode in the MDS, which should be correspondence with each other. Then we have to do calibration for distortion, our stage, and intensity range calibration to detect the hole on the quantifiable grid. And then we can submit our query for the final data collection. So earlier, um, then sir also said that why do we use ethane for plunging? Why not liquid nitrogen? So the basic answer is this: uh, because ethane is having high heat capacity. It means it requires more heat to uh, raise its temperature certain degree. When our sample dip inside the ethane, it releases heat. Instead of uh, boiling, it used to transfer that heat away from the R grid, and that generally leads to the vitrification of our sample. While in case of nitrogen, it is having very less heat capacity, which make it boil very early, and by getting some little bit of heat also. And another reason is that liquid nitrogen used to form a condensed layer or a very cold layer over it. And whenever we are dipping our sample inside the nitrogen, it may condense before plunging inside the nitrogen, which may lead to the formation of crystallize, and we can destroy our sample. Next, why the optimal ice thickness in a vitrified sample is important. Generally, the problem we are facing on day-to-day basis that it is very difficult to find the proper ice thickness. So for that, uh, proper ice thickness. So if ice is very thin, that it can break down during imaging or exclude out our sample of interest. And if our ice is too thick, then we are unable to see our particle of interest because it's used to scatter more electrons than transmit, and then can drift our sample more. That common challenges that we are facing is the preferred orientation. For that, we can optimize, not we can explore the preferred orientation by using the tilt series data collection. For example, if we are collecting 100 images, then better to collect 20 to 30 images on a certain tilt angle, or 70 to 80 on the normal. And some people used to also prefer the thick ice to deal with this issue. And another is how to tell the thickness of ice. So either we can increase blotting time, or we can decrease the sample volume, or we can increase number of blotting sheet. So here are some of the images that we collect in our facility. This is the filamentous negatively stained. These are the ribosome. Next one, negatively stained with PT. And these are the nanoparticles. Generally, metallic particle doesn't require staining, so we can visualize simply by putting our sample on the grid. After drying, we can image. Next. These are the ribosome micrograph that we collected in cryo mode. We are having 200 kV with the K2 detector. And next one, please. This micrograph is of clamp fragment, and these are micelles which somebody used to deliver their uh, drug, targeted drug delivery. Basically, they are using these micelles. So these are liposome, and the right images of apoferritin. Uh, so I would like to thank our executive director, Dr. Arvind Sahu, for providing such conducive and learning environment. Dr. Deepak Nayak, who is head of the ATPC, and Dr. Prem Singh Kaushal, who taught me how to run our sample, load our sample, and also the imaging part. And my colleague, especially Mr. Madhav Medikunda, who is also working in the same facility with me. Thank you. If anyone is having any query, then
Yep. Okay. Thank you, Dr. Reena, for such an exciting talk and showing us what is happening in the backstage of like what exactly, how much effort you are putting in purifying the protein of interest and getting the structures. So if we have audience, if you have any questions, this is the right time to pop it up. If you don't have any questions, then maybe we can go for a quick lunch and we can come back by 2.
Excuse me, sir.
Yeah, Dharmender, can you have a quick questions?
Yes, sir. Hi, Dr. Reena.
hi, Raminda.
If we have instructions, we can imagine the cryo-EM.
Yeah, we can do, but not get the intracellular detail. For that, we have to do cryosectioning.
Okay. Okay, if we have not the sample is not transparent, then we can use for cryo-EM, or we have to change the buffer of the cell.
Transparent means thick samples, or you are saying that. Actually, microscopy is all based on the transmission and blockage of our electron. Where our electron got transmitted, we receive signal. We have our path of our electron beam is blocked. We see.
Okay, thank you.
Any other queries?
Any other queries we have? Okay, so let us thank Dr. Reena once again for your insightful lecture on cryo sample preparation. Okay, thank you. We'll just close the session, the morning session for now. We will meet at 2 o'clock for another interesting series of talk. Okay,
Thank you.
thank you.
Thank you.